I know this week in April I'm going to die because I got this exam or something, you can look ahead. Yeah, and all the dates are updated in Canvas also. And since I posted all the other, like even the trivia points we do at the end, the song, the jam state, yeah, so if you want to, as it gets mid-semester onward, a lot of people start using that what if option to see, oh, would I need to get on this and this and this to get a B or an A or whatever. You can start planning ahead then. And then I sent you that Teams link yesterday also, so if there's any videos you want to look at from anything I play in class, keep that link handy somewhere, because that'll be what you go back to. So any video I have is in that Teams page. If it's a YouTube kind of video, it'll just be under post, you'll just see the topic and the slide number. And if it's a thing I've gotten from some book source or somewhere else where it's the whole video, then go to files at the top and then you'll see it's split up into unit 1, unit 2, and unit 3. Some people find that useful, some people have never used that in their lives, so they still do fine. Others are like, no, I need to go back and watch the videos, just see what you were talking about, or that one video was really useful that you played it in class, so I want to go back to it. That's really it. Lab itself will be most of the period again this week and next week, and then we start having a few shorter labs in there. But yeah, this week's lab is pretty important because we're doing gram staining for the first time, and that's the one method you actually have to get good at this semester. Everything else is like a one and done thing, but gram staining is kind of the thing you have to practice to actually get good at. I think that's everything. And I'll go over the 1.1 exam. If you want to see any of those findings, usually the best time is during in between classes after most people have finished their exam or quiz, or like after lab is another good time, or of course office hours. Anything else happening in your life? Good, bad? Somewhere in between. We're still alive, and 2 weeks are down. 17 more long weeks to go. We're out of it. So we are continuing with 1.3. And if I happen not to finish 1.3, whatever I don't finish is what I record on. It's like a Zoom recording and just send you the link afterwards. So then you can watch that. Usually if I do end up doing that, which happens a decent number of times throughout the semester, it's usually just a few slides, and we're mostly because we take our quizzes and exams at the end of lecture, so it's like 50 to 20 minute kind of thing. Yeah, someone was asking last time about like what's the axis when you're turning your flagella either clockwise or counterclockwise. It's going to be specifically based on where the attachment point is. Like if it's a peritrichous bacterium where it has flagella all over its body, it's based on wherever it's anchored to. So even if it's not at a pole, it's based on whatever side of the bacteria it's anchored to. If it's going counterclockwise, it's moving forward. If it's going clockwise, it's jumping. But it's looking down on wherever it's anchored to. I think we played this and did this slide last. So this is where we left off last time. So we said counterclockwise rotation is moving forward, counterclockwise is tumbling, and then positive chemotaxis is going to something you want to go towards in the bacterium. Imagine going to like Taco Bell is going in that direction because you want to, versus something negative. It's like eating peas and broccoli, you want to go away from that. So you can actually visualize this in a liquid or a semi-solid medium. Like if you put something, a attractant, like you have this slide, if you put an attractant in there, things are going to go towards the straw. If it's a repellent, things will go away from the straw. So also related to movement are specialized flagella called axial filaments. And these, they have the same rules of like counterclockwise versus clockwise. They're only unique because of where they're actually located relative to the cell. So there's gram-negative bacteria. You got an introduction to those last time where we said they have an outer membrane. And in the coming slides, we'll go through more about positive versus negative. But axial filaments, the key is that they're actually inside of that outer membrane. So they're also called endoflagella because they're internal, they're inside a membrane. They're found specifically in those bacteria we talked about earlier when we talked about bacterial shape, the ones that have multiple twists, so the spirochetes. And they're in the periplasmic space, in those spirochetes. They're anchored on only one end of the cell, and then the flagella run all along the length of the body of the bacterium. And as they rotate, then it's going to cause the cell to move like a corkscrew as it's going through a liquid. So if you look here at that, if you look along the length of this bacterium, they'll be anchored on one end, but then they go all along the length of it. So they're still spinning around, but they're inside of a membrane. All the movements we've talked about so far, they're spinning around outside of a membrane. So we play a video of this actually. You can also see how they move in a corkscrew fashion when they move. It's under special microscopy where you actually stain the bacteria. Then you can see what they actually move like. So you can see they're spinning around through the liquid. That's because there's the flagella all along the length of their body as they're spinning. Wow, that's so cool. I can watch spinning bacteria for hours. Yeah, I know, it's pretty relaxing. I've given the analogy before, like think about if you've gone camping and you're in a sleeping bag, but you had a lot to drink the night before, and then the next morning or during the night you're trying to get out of that sleeping bag, but you don't know how, so you just kind of keep spinning around in the sleeping bag. That's sort of like how these bacteria are moving around. So think of that sleeping bag as the outer membrane, and your arms and legs are waving around, but you're still inside of that sleeping bag. Strangely, a lot of micro students can relate to that. I don't know what kind of cool camping stories you have, but there's a lot that can. So if we watch a video of these in action also.
Spirochetes are a group of bacteria that are spiral-shaped. They move in a corkscrew-like fashion, using flagella that do not protrude from the cell wall like typical flagella. Lyme disease is caused by a spirochete called Borrelia burgdorferi. Another spirochete, Treponema pallidum, causes syphilis. Spirochetes have an axial filament that lies between the outer membrane and the cell membrane. The axial filament winds around the outer edge of the cell. The axial filament is made up of a bundle of modified flagella called endoflagella. Rotation of the endoflagella causes the axial filament to rotate around the bacterium, which causes the spirochete to corkscrew through its medium. It is thought that this manner of movement allows the spirochetes to move more easily through viscous human tissues and fluids, such as mucus. Then they can easily attach to the cells below.
It's a great way to spin around, especially in a liquid. Even if it's a thicker liquid like the mucus that a lot of our cells produce, you can still spin through that. Whereas if it's something that doesn't have that ability, it gets trapped in the mucus and then our immune system can clear it out. So visually, if you look at these, you can see how they're oriented relative to the bacteria. And you'll only find these in gram-negative bacteria because they're the only ones that have this outer membrane. It's a gram-positive, if you remember when we did a quick intro to that, they just have a thick cell wall on the outside. So there's not enough room for a flagellum inside of that cell wall. Whereas in gram-negatives, where they have this thin cell wall and then they have an outer membrane outside of that, you can have these flagella. So this is just under that outer membrane, but it wraps around the whole body. And as it rotates, it propels the cell sideways rotating. Then going into other projections from a bacterial cell. So then you also have fimbriae. So these are all of these little projections. There's usually a lot of them on the cell if it has it. They're hair or brush-like appendages, and they're going to allow for attachment. They're not for motility, but they're to help to stick to a surface. Like when you don't brush your teeth for a couple of days and you feel that sliminess, a lot of that is because of fimbriae. All along your intestinal walls, like you have natural bacteria that stick to the intestine. So even though you're eating and drinking, they're not just washing away with that liquid. That's largely because of these fimbriae that help to stick. They're more common than the other projections we're going to talk about, which are the pili. So there'll be a lot more fimbriae than there are pili. The pili, on the other hand, are involved in some types of motility, particularly called gliding and twitching motility. This is a unique kind of motility that only a few bacterial species have. It's sort of like if you've done any rock climbing where you do the grappling hooks and you're going up the side of a mountain and latch onto the side and then pull yourself, and then once you're pulled to that next motion, then you throw the hook again and then you pull yourself to the next position. That's sort of like how pili do. So they're not rotating, they're kind of acting as hooks in the side of something so they can move in twitches. They'll move a little, stop, reposition themselves, throw the hooks out again, hook on, and then move again. There's also other types of pili known as conjugation pili. And these are involved in a process of DNA transfer from one cell to another. We'll talk more about this in Unit 2 when we talk about conjugation as a way to transfer DNA from one cell to another. But say you have one bacterium that's resistant to penicillin or tetracycline or some antibiotic, it can transfer that resistance gene to a different bacterium that isn't resistant. And it can do that by a physical bridge, which is one of these pili. So there's a mating process of bacterial cells that, even though there's not male and female bacteria, but this is the closest they get, one bacterium will stick out a pilus and then transfer some of its DNA, like resistance to some antibiotic, from one cell to another. So it's a physical bridge from this cell to this next cell. And that's how you have a donor cell and you have a recipient cell, and the recipient cell gets that DNA. Then going inward, we have the bacterial cell wall. So both gram-positive and gram-negative bacteria are going to have that. And the timing works out nicely because we're talking about the theory of what the cell wall is like in lecture, and then this week's lab, we're doing gram staining, which is purely based on that cell wall and outer membrane. So when you have the cell wall, it's going to prevent osmotic lysis. So the cell, it's usually a tougher structure than a membrane. So it's not just going to burst open or shrink inward. Even if you change a bacterium from, say, fresh water to salt water or salt water to fresh water, it's going to stay in a relatively similar shape. And it also physically protects the cell membrane that is further inside of the cell wall. It's made of a molecule called peptidoglycan in bacteria. There's other things that have cell walls that are made up of different molecules, like plants have cell walls, but they're made up of something else besides peptidoglycan. When we talk about disease in Unit 3, that cell wall is also going to be important because it contributes to pathogenicity. It triggers your immune system. Sometimes it can cause an overactive immune response, and you can actually die from the inflammation associated with a bunch of bacterial cell walls in your bloodstream. So relative to where the bacterial cell is, on the outside you have the cell wall. Inside of that you have the plasma or cell membrane. And then other structures we're going to be talking about. We already talked about the capsule, the thick thing that's secreted by some bacterial cells. That's on the outside of the cell wall. And then inside of that, you'll have the DNA in a region of the cell called the nucleoid region. And then you'll also have some protein-making structures called ribosomes. And they're the same, they have the same function. If you've taken physio, you've heard about ribosomes in eukaryotic cells. It's the same thing, except they're in bacterial cells. So the cell wall itself, it's made of peptidoglycan. It's a polymer of repeating disaccharide. Who can tell me what a saccharide is?
Sugar.
Yes, a sugar. So if it's a disaccharide, it's two sugar units together, but they're in rows. And those rows are of two different sugars. You have N-acetylglucosamine and N-acetylmuramic acid. So peptidoglycan itself is just that alternating. It'll be nag, nam, like that over and over again. The rows then are linked by small peptides, which are just small sequences of amino acids, usually 4 or 5 amino acids between them. So it's a tiny bunch of sticks together with row. Each one of the sticks is this alternating N-acetylglucosamine and N-acetylmuramic acid, and then in between them, the rope that's tying those sticks together are these polypeptides. You obviously don't need to know this exact structure or anything. Save that for a chem course. But there's bonds between the NAG and the NAM that hold them together like that. And these bonds that hold those together are sometimes targets of certain antibiotics. If they're the kind that affect cell wall synthesis, they'll break these bonds. So when the cell tries to reproduce or replicate, the cell wall can't expand with that, and it breaks apart and the cell dies. So now if we look at it row by row by row, so alternating NAG and NAM, just all along here, the whole length, and then you have these small polypeptides of a few amino acids that are linking the rows together. And those are often the targets of the different antibiotics, is to break not necessarily the NAG-NAM held together, but often to break these small polypeptides. And that way when the rows are expanding or need to expand, they don't hold together. Just gonna walk out on me.
No, I'm not.
Okay, sure.
I just need to grab something.
Sure. That's what they all say when they walk out on me and never come back. So these are those small the polypeptides are called cross bridges. So when an antibiotic targets those, they'll say something like, oh, it targets cross bridge formation. It's targeting these. So it doesn't necessarily break those either. It might just weaken them so that if the cell needs to make more peptidoglycan, that's when you won't be able to hold those rows together. So now, this will be important when we go back to staining this week with gram staining. So you'll see this exact diagram again, either this afternoon or on Wednesday afternoon. But when we do gram staining, it's based on these structural differences between the two types of bacteria. And most bacteria can be classified as either gram-positive or gram-negative. There are a few species of bacteria that just don't have a cell wall, so they don't go through gram staining. But most bacteria out there, whether they make us sick or not, can be classified as gram-positive or gram-negative. So both of them are going to have a cell membrane or plasma membrane in yellow there. But then everything outside of that is what's different. So if it's a gram-positive bacterium, it has one periplasmic space. That's just the space between a wall and membrane or two membranes. So that's a space in between here. And then outside of that, it has a very thick cell wall. That's this brown layer here. And that's because it's not just one sheet of peptidoglycan, it's sheet upon sheet upon sheet stacked up, and that's what makes it a very thick cell wall. So when we talk about staining this week, a big reason certain bacteria stain certain colors when they're gram-positive is because of that thick cell wall is going to keep the dye inside the cell, so it appears one color. When we talk about gram-negatives then, they also have the cell membrane, but then they're going to have two periplasmic spaces. So the first is between the cell membrane and a thin cell wall in purple there. And then there's a second periplasmic space between the cell wall, which is very thin, and an outer membrane. That's the thing we just saw those axial filaments within. That's within that green outer membrane. And that's only going to be in the gram-negative bacteria. So on these, because they have a very thin cell wall, it's easier to remove dyes from them when you go through the staining process. So gram-positives will retain a dye called crystal violet because they have that thick cell wall. Gram-negatives will lose that
purple color, they'll lose the crystal violet because they have a thin cell wall, so the dye gets decolorized or removed from the bacteria. So with gram-positives, they have a very thick peptidoglycan layer. There's only one periplasmic space. It's between the cell wall and the cell membrane. They also have teichoic acids. So there's lipoteichoic acids. Those are going to be the things that link the cell wall to the plasma membrane. Something has to anchor that thick cell wall to the membrane. Otherwise, the cell wall would just keep spinning around the membrane. Then there's separate acids called wall teichoic acids. They're the ones that are going to link the peptidoglycan layers because it's layer upon layer upon layer that makes it a thick cell wall. These teichoic acids have a negative charge to them, and that's important for various drugs. If you want to get a drug through there, it has to be something that's small and oppositely charged, so it can actually be attracted to that wall. These cell walls of the teichoic acids will also regulate movement of cations because of that charge difference. So positively charged ions are called cations, and they're attracted to negatively charged things like these acids in the walls. Versus if you exposed it to some negatively charged drug, it wouldn't even get close to the bacterial cell because like repels like. That's what they say about finding love and relationships and opposites attracting. That's the same thing here. Positive and negative are going to be attracted. Negative and negative are going to be repelled. So someone's like, no, but I'm exactly like my partner and we still love each other after years. And then someone else chimes in, no, but I was so much like my partner. We're always fighting every day. I've had those conversations in micro classes. It's a bonding experience or commiserating depending on your relationship history. So we zoom in on this then. Here's our bacterial cell. It's a gram-positive cell. Do a cross-section of these layers here. So you have the plasma or cell membrane. Then you have the thick cell wall. So lots of rows of peptidoglycan side by side. They're held together by those short polypeptides. And then they're stacked one on top of the other also. So there's the two types of teichoic acids. The lipoteichoic acid is going to anchor the whole cell wall to the cell membrane, so it doesn't just keep flopping or spinning around. And then the wall teichoic acids aren't tied to the membrane, they're just tied to the individual layers on top and bottom. So that keeps that thick cell wall anchored to the actual cell itself. And that's going to be very important because when we do staining, if you put in a dye, if you can get it in, then it's going to be hard to get it out because there's this thick cell wall. It has to go basically through a really thick, porous substance to come back out. In gram-negatives, on the other hand, they have a thin peptidoglycan layer. Often it's just one layer thick. It also is going to have an outer membrane outside of that cell wall. And that's made of polysaccharides, lipoproteins, and phospholipids. So polysaccharides are the sugars, lipoproteins are combinations of lipids and proteins, and then phospholipids are phosphate groups attached to lipids. In gram-negatives is where you'll find lipopolysaccharides, or LPS, and the outside of that layer in the outer membrane is known as lipid A. That's an endotoxin. It's going to be something that stimulates inflammation in us when we have gram-negative infections. That's often why they can only use certain antibiotics with gram-negative infections, because if you killed all the cells all at once with an antibiotic, they would release all that lipid A as the cell breaks up and dies. And that actually overstimulates your immune system to where you can die of endotoxic shock. If you've worked in a clinical setting, you may have heard of a patient going into endotoxic shock. That's what that is. They had a gram-negative infection that overstimulated their immune response. Then, as we saw in that image, there's two periplasmic spaces, one between the plasma membrane and the cell wall, the second between the cell wall and that outer membrane. And between those two is that peptidoglycan layer. So it's between the outer membrane and the cell or plasma membrane. Then you have the porins, and these are specific proteins that form channels through the membrane. So in gram-negative, a lot of times it's hard to get drugs or antibiotics inside there because they can't go through that outer membrane. You have to make something or find a drug or antibiotic that can specifically go through those porin channels. So if you do a cross-section of a gram-negative then, zooming in on this layer here, now you have the plasma membrane down here, cell membrane. Then you have the first periplasmic space. Then you have the thin cell wall. It's only one layer of peptidoglycan. Then you have the second periplasmic space between that thin cell wall and the outer membrane. And then you have the outer membrane. And the outside of that outer membrane is where you would find lipid A. That's the part that stimulates your immune system. And then if you zoom in here, there's also some phospholipids in there, so phosphates attached to lipids. There's the lipoproteins. Then you'll have some of these porin proteins that are channels. So a lot of times, only certain drugs can get into gram-negatives because they're designed to go through those porin channels. So when we do gram staining, like we're doing this week in lab, you have multiple reagents that you're using, and you'll do just a regular run through of it today. But after that, it's just practicing the timing and which reagents you want to use and how much you apply and all of that. And that just comes from going through the process over and over. But crystal violet is your first reagent. It's a dye. It's going to bind to your second reagent, which is iodine. And that forms crystals together inside the cell. So crystal violet with iodine is a larger molecule or structure than just crystal violet by itself. So once you add the iodine, it forms these larger crystals. So now it's going to be harder to lose crystal violet from the cell if it's bound to iodine. Now when you do gram-positive versus gram-negative cells, you add ethanol or acid alcohol. That's your decolorizer. It's going to dehydrate that thick peptidoglycan layer. Now it's sort of like a dry shell on a bean or pea pod or something like that. So now the crystal violet iodine crystals are trapped inside that thick cell wall. So that purple color that comes from that dye is going to remain in those cells. In gram-negatives, when you add those decolorizers, it's going to dissolve the outer membrane, and there's holes left in that peptidoglycan layer because it's only one cell, one sheet thick. In gram-positives, it was really thick, so it was harder for things to get out. In gram-negatives, you just have that one layer. So now you still form the crystal violet iodine complexes, but now it's easier to wash it out of there. So they lose that purple color. Then when you add safranin, which is your second dye, it adds color to both the positives and the negatives, but the positives still appear purple because purple is darker than the pink that you see of safranin. In gram-negatives, you're going to lose the purple completely, so you only see the pink of the safranin. So you see differences like this. So gram-negative bacteria, when you do the staining, should appear pink like this. Gram-positive cells should appear purple, even though you're exposing them to all the same things for the same times. It's the specific properties of the bacteria that give them this different appearance. Then continuing inward, we have the plasma or cytoplasmic membrane. And this is one of those things we said is common to all cells, whether it's our cells or bacterial cells, they're all going to have a plasma membrane. So it's always a phospholipid bilayer. That means there's phospholipids on the top and phospholipids on the bottom. That's what makes it a bilayer. And that is going to enclose within it the cytoplasm, which is the liquid portion inside the membrane. There's peripheral proteins that are on the periphery, so they either stick out the top portion of that bilayer or the bottom portion, but not both. And then there's integral or transmembrane proteins that go all the way through the membrane. So they have portions sticking out from the top and from the bottom. If you zoom in on a bacterial cell, so it's a lipid bilayer. Outside of that, you have the peptidoglycan, and outside of that, if it's a gram-negative, you also have the outer membrane, and that's also a bilayer. So cross section of that zoomed in now, when we talk about phospholipids, there's a phosphate group that's going to be part of the polar head that has some charge to it. So it's aqueous. It doesn't mind interacting with water outside of the cell or inside of the cell because it has some charge to it. Inside of that are the nonpolar fatty acid tails. They're like the oils, and water and oil don't mix. So in this case, oils try to congregate. The fatty acids are attracted to each other and they want to be as far away from the water as possible. So if you do like vinegar salad dressing, you always have to constantly shake it up to keep it together because pretty quickly, as soon as you put the bottle down, the oil and the vinegar separate. That's the same thing here. So imagine on the top is water, on the inside is water, in between is oil. So if you just threw a bunch of these polar heads and fatty acid tails together, they'll naturally form a lipid bilayer. Even if it's not a living cell, you see these natural bilayers form because the fatty acids are attracted to each other and the polar heads want to stay towards the water. And then on the periphery, either from one of the two layers, an inside or outside, will be these peripheral proteins, and then some of those transmembrane or integral proteins will go all the way through this bilayer. So let's play a video of this. Most of us are still awake. That's a good sign. The true guests of this is survive unit 2. But that's not as much micro. It's still micro, but it's more concepts and chemistry and chemical structures and all that good stuff.
The cytoplasmic membrane of a prokaryotic cell is the main gateway for movement of molecules in and out of the cell. This membrane may also be called the cell membrane or plasma membrane. Cytoplasmic membranes in bacteria are comprised of a phospholipid bilayer. Each phospholipid contains a phosphate head located on the outer part of the bilayer, attached to a glycerol and 2-fatty acid, which face the core of the membrane. The phosphate is negatively charged and hydrophilic, while the tail portion of the phospholipids is hydrophobic. Thus, cytoplasmic membranes are very effective at blocking passage of many molecules, while other molecules are allowed to pass. Cytoplasmic membranes also contain 2 types of proteins. Integral proteins often span the entire bilayer and act as channels for transport of molecules that normally wouldn't be able to penetrate the bilayer. Peripheral proteins are loosely attached to the membrane on one side or the other. Some of these may be glycoproteins, which are proteins bound to a polysaccharide. These proteins may function as recognition sites, enzymes, or receptors.
So what does hydrophilic mean?
Attracted to water. Yeah.
What about it, water?
It likes water.
Yes, so it's going to be attracted to water. And hydrophobic?
It doesn't like water.
Yeah, so all these lipid tails in between here are going to be hydrophobic. They're afraid of the water. The polar heads, the glycerol side of these phospholipids, are going to be hydrophilic. They love to interact with water. Which brings us to the fluid mosaic model of bilayers. And this is true for our cells also. It's not just a micro thing. But there's the general idea of how biological membranes are created or set up. So based on that model, you have a membrane that's viscous as olive oil. If you think of how thick olive oil is when you pour it on something or use it in cooking. So that's about how viscous or thick a membrane is. Then there's proteins that are going to move freely for various functions along that bilayer. So if there's a protein that's within that phospholipid bilayer, it's not just fixed in that one position, it can move around. And it usually is moving sideways. So it might be, say if you're thinking GPS units or something, so at one point that protein is at position XYZ1. At 2 seconds later, it can be at position XYZ2 because it can move sideways along that bilayer. So phospholipids, on the other hand, they can rotate like top to bottom or bottom to top, and they can also move laterally, so they'll move side to side. It's also self-sealing. So again, think of olive oil and you pour some in on a plate or something, and then you stick your finger in there. You create an indentation, and as soon as you pull your finger out, the oil closes that up. That's the same way these phospholipids work. Plasma membrane is selectively permeable, so it's not just going to let anything through there. Certain things can pass through there, other things can't. And that's a good thing for the cell. The cell isn't just an open book saying, yeah, come on in or go ahead and leave, I don't care. It cares about what can get into the cell and what doesn't. Also, you'll have enzymes in that membrane for ATP production. When you get to unit 2 or topic 2.2, you'll hear way more about ATP and you'll hate it with a passion. But ATP is going to be the currency for cellular energy. So it's going to be what our cells have to make lots of, it's what bacterial cells have to make lots of. But in our cells, those enzymes that help to make ATP are located in different regions than in bacterial cells. So if we play a couple of videos on this and we think about membrane permeability, what does it take to actually get into or out of a phospholipid bilayer? Because not everything can get in or come out of the cell.
With their hydrophobic cores and charged surfaces, cell membranes are very efficient at blocking passage of many molecules. Some molecules, such as water, dissolved oxygen, and simple alcohols, are able to pass through cell membranes freely by passive diffusion. However, charged molecules and large molecules cannot pass directly through cell membranes, and hydrophobic molecules tend to become stuck inside the hydrophobic interior of cell membranes. In order to allow these complex molecules to enter and exit the cell, organisms rely on two forms of membrane transport: passive transport and active transport. Passive transport occurs along an electrochemical gradient. It does not require the expenditure of energy in the form of ATP for transport. Active transport requires the expenditure of ATP in order to transport molecules across the membrane. Active transport and some forms of passive transport require the use of transport proteins, such as the integral protein seen here.
So some things can just go straight through the phospholipid part. Some things can only go through these protein parts because they're too big or too charged or something about them doesn't allow them to go just straight through this phospholipid part. And as we talk more about energy requirements, so if it's passive transport, it doesn't need energy to go through a membrane. If it's active transport, it actually needs energy. So there'll be some of you like on quizzes and exams, oh, I only looked at the material once and I got a perfect score on it. And there's others like, I ran over that 10 times and it still took me so much to just pass the quiz or exam. That's like an active transport. You're taking a lot of energy to get through the test, versus the passive transport is like, I just watched this lecture one time and I got a perfect score. So passive processes are always going to be where substances are moving from high concentration to low concentration, and that doesn't take energy. Things want to go from high concentration to low concentration. So in living things, if they're going across a membrane, if you have something that's really high concentration on one side and low concentration on the other, things will want to go overall from high to low. That doesn't take energy. Things will naturally go through that. Active processes, on the other hand, move from low concentration to high concentration, and that takes energy. So if you have like a closet at your apartment or house or wherever you live, and you're always throwing random stuff in there, once it gets packed and you're still trying to squeeze more in there, it's like, oh, I just throw it in there. I don't care where it's at. I just want to fit it in that closet. It takes more energy as you fill that closet up more. And at some point, of course, then when there's a lot of stuff in that closet, you open the door and everything falls out of there. That's passive transport. That doesn't take energy because it's going from a high concentration of crap that you threw in the closet to low concentration on the floor outside of that closet. So example of passive processes, you have simple diffusion. That's where you have movement of a solute. A solute is something dissolved in a solvent, a liquid. So simple diffusion, you have movement of a solute from an area of high concentration to area of low concentration, and that continues until you reach a point called equilibrium. So if you have a plasma membrane like this, there's a lot of these reddish colored balls on the outside and a few on the inside. So high concentration of solutes here, low concentration here. General movement will be high to low across this membrane, and that will continue until the concentration is the same on both sides. So that doesn't mean that once you reach equilibrium, movement stops. When you're at equilibrium, that just means that as many as are going from top to bottom, there'll be that many going bottom to top. If we watch a video of that, there's different types of diffusion. Diffusion is always going to be passive, so there's no energy required, but there could be other things required. So facilitated diffusion, as this video is going to show, is where you have some protein involved that helps the diffusion process. Simple diffusion is just going across the phospholipid layer like this. This video will show when you have non-specific facilitated diffusion, maybe it's just going to allow all positively charged molecules to come through, but it has to come through this protein. It's still going high to low, so you still don't need energy, but it can only happen through this protein. When we talk about specific facilitated diffusion, you'll see that only one specific molecule can come through, maybe only this green one. This red triangle may be similar charge or shape, but it's not that specific molecule, and this purple protein is only meant to allow the green molecule to go through. And then lastly, when we get to osmosis, osmosis is just passive diffusion, but of water. So you have to think of water as the thing going from high to low. So let's watch the video.
Passive transport includes diffusion, non-specific and specific facilitated diffusion, and osmosis. All of these processes occur along electrochemical gradients and do not require the expenditure of energy in the form of ATP. Diffusion is the net movement of a chemical down its concentration gradient, that is, from an area of high concentration to an area of lower concentration. This diffusion requires no energy expenditure by the cell, as the concentration gradient drives the diffusion. Small uncharged molecules, such as dissolved oxygen, dissolved carbon dioxide, small alcohols, and small fatty acids, can freely diffuse through the cytoplasmic membrane. Once equilibrium is reached, molecules will still move across the membrane, but there is no net movement in a particular direction.
But wait, there's more. So facilitated diffusion, on the other hand, you're not going through just a phospholipid bilayer, you're going through a specific protein. So here you have the solute that's going to combine with a transporter membrane or transporter protein in that membrane. It's still going high to low, so it still doesn't require energy, but it requires going through that protein. Maybe it's just too big to go through the phospholipid bilayer, but it's still going high to low. So it's often a transport of ions that have some charge to them. Maybe there's a whole bunch of positives on this side and just a few on this side. So it's still a high positive charge to low. Or just the size of the molecules. It's too big to go through the bilayer, but it can go through this protein. But it's happening with the concentration gradient. So the gradient is always across a membrane, and it's always whether it's high on one side and low on the other. So when I say things like, oh, it's going against a concentration gradient, that means you're going from the low concentration to the high concentration. So if we watch a video covering the facilitated side.
Diffusion does not require special membrane proteins for movement of molecules. However, other types of diffusion do require such proteins for the transport of molecules. Non-specific facilitated diffusion is passive transport allowed by transmembrane proteins called permeases. In the case of non-specific facilitated diffusion, These permeases allow a wide range of molecules that meet charge or size criteria to pass through their channel. Some facilitated diffusion is passive transport allowed by transmembrane proteins called permeases. In the case of a specific facilitated diffusion, these permeases only allow specific types of molecules to pass through their channel. The channel contains a recognition element. If the molecule is not recognized, the channel will not open to allow the molecule to pass into the cell. Osmosis is a special kind of passive diffusion that involves the movement of water molecules across a semi-permeable membrane. Water passes through the phospholipid bilayer with some difficulty, but transmembrane channels can ease the passage of water through the membrane in a process called facilitated diffusion. Water plays a critical role in maintaining cell integrity. Water tends to move across the membrane from areas of high concentration to areas of low concentration, just like solutes. The difference is that water makes up most of the liquid, so a change in the total concentration of solutes is really the force that causes water to move. If the concentration of solutes outside the cell is high compared to the concentration inside the cell, water may leave the cell, causing the cell to shrivel up. Similarly, if the concentration of solutes inside the cell is high relative to the outside, water may move into the cell and can cause it to swell up and possibly burst.
And this is a great video to go back and watch later on if you're confused on how that works and which direction things move. So osmosis is the one that usually trips up students because it's still movement from high to low, but it's movement of water, not of the solutes that are dissolved in the water. So osmosis is specifically the movement of water across a permeable membrane from an area of higher water concentration to an area of lower water concentration. So it's still through a lipid bilayer or through specific proteins that can travel, allow transport of water molecules that's called aquaporin. But if you think about what's dissolved in there, so if the concentration of solutes is high, the concentration of water is going to be low, and vice versa. So if you have a whole bunch of salt dissolved in water, there's more salt there, so there's less water. If the concentration of salt dissolved in water is low, the concentration of water is high, because there's more water and less salt. So you can see osmosis in action using one of these kind of experiments. Some of the Chem 20 classes here do this kind of thing. But say you put a sugar in a cellophane sack like this, like sucrose is a sugar. So you put some of that sugar in the form of these purple balls inside this bag. It's semi-permeable, so the sugar can't leave the cellophane sack, but water can go in and out of it. You put this in with a stopper tube like this, and there's a glass tube that goes out of the beaker. And then on the outside, you just have pure water, there's no sugar. So initially, when osmosis happens, so the sugar can't leave the sac, water can go into the sac. So there's more water out here because there's no sugar dissolved in it, and there's less water in here because there is sugar dissolved. So water is going from high concentration to low. It's still a passive process, and it fills up this sac and actually raises the level of liquid in this tube. So you start off with this level here at the top, the same as this. After a while, this level goes way above the level of the liquid because water is entering the sac. It's trying to even out the concentration. So at this point, water is at equilibrium also. And it went from high concentration of water to low concentration. Related to that, then you can come up with two different types of solutions based on what you put a cell into. So there's isotonic solutions. That's where the solute concentration is going to be equal inside and outside of the cell. So that way, a cell is put in an isotonic solution. That means the liquid that's inside the cell, the solutes that are dissolved in that are at the same concentration as the liquid you just put the cell into. So net movement is zero when you put a cell in an isotonic solution. Hypotonic solutions, the solute concentration is lower outside the cell than inside the cell. So based on water going from high to low, water will go into the cell. It's going in the opposite direction of what the solutes are. So that means there's a whole lot of solutes inside the cell compared to the outside, opposite of water, which is high on the outside and low on the inside. So water moves into the cell, down its concentration gradient. Opposite happens in hypertonic solutions, think salty solutions. So you put a bacterial cell in a salt solution where it's really high salt, solute concentration now is higher outside of the cell than inside. That means water concentration is higher inside than outside. Water is going to move down its concentration gradient, which means water leaves the cell and goes into the solution, and the cell shrinks. So if you visually see this, isotonic, there's no net movement of water because solute concentration is the same inside the cell as outside of the cell. There's still movement, but it's equal. So just as many solutes leave as enter, or just as much water leaves as enters. If you put a cell in a hypotonic solution, water is going to enter the cell because the solute concentration is higher inside the cell than outside. So the cell will eventually swell up and burst open. If you put the cell in a hypertonic solution, there's more solutes outside the cell, which means there's less water outside the cell. Always think solutes versus water are going to be opposite. If it's high solute, it's low water concentration. So in this case, then water leaves the cell, goes into the surrounding environment, and the cell is going to shrink. With active transport, you require energy, and that energy is going to be in the form of a molecule called ATP, and also you'll have a transporter protein through which transport can occur. When you have active transport, you're going against a concentration gradient. So now you're going from low concentration to high concentration. If it's just standard active transport, it doesn't alter whatever's passaging as it's crossing. You're adding energy, but you're not changing the form of whatever ion or molecule is going across the membrane. Another active process is group translocation, and this requires a transporter protein and something called PEP, or phosphoenolpyruvic acid, for the energy source. This is going to go against the gradient because it's active, but the substance that's going across the membrane is also altered in the process. So on one side, it's some chemical structure. By the time it crosses the membrane, it's a different chemical structure. And that's part of it. So it requires energy because it's going from low to high, but it also needs to change its shape around. So we play this video, which is a great stopping point after this video. So this mind-blowing stuff. Things that dreams are made of.
Active transport requires the cell to expend energy, usually in the form of ATP, to transport materials across the cytoplasmic membrane. This transport occurs through transport proteins and is usually against the electrochemical gradient, like carrying a load uphill. There are three types of transport protein. Uniport proteins transport only one type of molecule. Antiport proteins transport two types of molecules, but in opposite directions. Symport proteins transport two different molecules at the same time, but is coupled with a uniport channel. A uniport is an active transport protein that transports a single molecule or a group of the same molecules in one direction against the concentration gradient. These proteins are also ATPases, using ATP to drive the transport. An antiport is an active transport protein that transports two molecules in opposite directions against their concentration gradients. These proteins are also ATPases, using ATP to drive the transport. An example of an antiport protein channel is a sodium-potassium gated channel that maintains the electrochemical gradient of cells, which is responsible for maintaining a voltage difference between the outside and the inside of the cell. A symport is an active transport protein that transports two different molecules across the cell membrane at the same time. In this case, it's coupled with a uniport channel that uses ATP to actively transport a molecule against its concentration gradient. When those same molecules are in high concentration outside the cell, their diffusion back into the cell provides energy to transport a different molecule against its concentration gradient through the symport.
So active processes, all this require energy, because they're all this somehow transporting something from low concentration to high concentration. So I'll finish off these last 8 slides or so as a recording later tonight or tomorrow at some point. Questions?